Hi everyone, so this is our third video on sex determination, and we're going to talk about how do new forms of genetic sex determination evolve. Okay, so in our last two videos we've learned that vertebrates share this core switch consisting of 3 genes, but that what flips this switch from the male state to the female state varies across different groups of vertebrates. So in this video, we're going to try to take this further and deeper in terms of our understanding, not just saying, okay, these genes do what we're told they do, but to kind of understand why they do and why we see some similarities even amongst the differences. So the first thing you might have noticed is that a lot of these genes that are involved have the same DM in them. And the similarities would seem even more striking if I had told you that SOX9 means the SRY box 9. Right, so all the genes that are serving as the gene that flips the switch are related, right? These 4 are related to each other, and then this one is related to one in the core switch. So while I've told you that these are independent, they're on different chromosomes, there seems to be some similarity. So in this video, we're going to try to understand a little bit more about why. Okay, so I thought we would stimulate this by imagining some mutations. So what I've shown here is the core switch plus this one gene that pops up a lot. DMRT1 seems to have been present in the ancestor of all vertebrates, and it plays a large role in a lot of modern vertebrates. And so if we start out with the case where the environment is regulating DMRT1, sorry that I used this block for it. The environment isn't blocking DMRT1. That this should more just be a line. It's regulating DMRT1, right? So DMRT1 is being regulated by the environment, and then based on its expression or not, possibly stimulating SOX9. Okay, so let's imagine starting with this circuit. Let's imagine some mutations that can occur. Well, one thing that we could imagine is a mutation in our regulatory site. So DMRT1 is somehow being regulated. One possibility is that it's being regulated at the level of transcription. If that were the case, and if it were a negative binding site, then what we can imagine is that if this site is mutated, then DMRT1 could become insensitive to the environment. Right, that regardless of the environmental cues, that DMRT1 could be expressed. There are also other options that we could imagine. We could imagine a change in the protein coding sequence that would give us something more similar to the super repressor that we saw when we studied the lac operon. So let's call this a super stimulator. That is, it's going to be able to stimulate SOX9 whether or not, regardless of what the environmental cues are, right? Because it's going to always be transcribed, not repressed, and so stimulating SOX9. Okay, so let's think about what would happen. So if we had an individual that was heterozygous for the wild type allele and the super stimulator allele, what would we expect to see? Maybe a good way to start to deal with these things would be to think about the case of a wild type in the case of a diploid. And so here we have our 2 different alleles. Okay, so in the case of a diploid and of a wild type diploid, they're both being regulated by the environment, so they're going to have the expected behavior. But what if one of our alleles, as in this genotype, is a super stimulator? Well, we said that this one is not going to be repressed by the environment, and so it's always going to be on. So this individual we expect to always be expressing DMRT1, which is going to lead to expression of SOX9 and FGF9 and little expression of Wnt4, which is to say that individuals that carry even one copy of this gene are expected to express the male regulatory program. Well, let's think about a different kind of mutation. So what if we had a mutation in DMRT1 such that it didn't function? So that if this is just knocked out overall, then it will never express DMRT1, so we'll never stimulate SOX9. So again, we could ask, well, what do we expect in the case of different diploid genotypes? So here are our two genotypes again. So here we're saying that the one on the bottom is a knockout allele of DMRT1. If that's the case, then we're going to get no stimulation from our bottom allele and only get stimulation from our top allele. So that means SOX9 is going to get less stimulation than it otherwise would. And so this may lead to a female genotype. Sorry, a female regulatory program. So it may be the case that a single mutant copy of DMRT1 would lead to a female regulatory program, a female development, regardless of the cues from the environment. But we could also think about a case where we have two mutant copies of DMRT1, and in those cases we would definitely expect little stimulation of SOX9 and therefore a female regulatory program. Okay, so what does this have to do with anything? Well, another question that we're asking here is, what is a sex chromosome? Where do sex chromosomes come from? How did they evolve? What do we mean when we say a sex chromosome? So let's talk this through. So a sex chromosome, right, is a chromosome for which the complement of chromosomes determines physical sexual development. So, for instance, in some cases, it's a question of whether you're homozygous for an X chromosome, a particular chromosome, in which case you're female, or heterozygous for an X and a Y, in which case you're male. Or in the case of a ZW system, it's a question of whether you're homozygous for a Z, two Zs, or whether you're heterozygous for a Z versus a W, female. So what have we been talking about in this video? So we've been talking about genes for which the complementation of your alleles might determine sex. So in our first case, we talked about that, sorry, not our first case, but one case we talked about was a DMRTY minus, DMRT1 minus, and we said that in that case, if you're homozygous for it, then we would expect you to develop as a female. So it's sort of the exact same thing that we were thinking about for X chromosomes. Suddenly, we've said that for a single mutation, a single deactivating mutation, could lead to the same thing, that if you're homozygous for it, we would expect female development. The other one, we also talked about a mutant where a super stimulator mutant, that even if you were heterozygous for it, we would expect male development, just as with an X and Y chromosome. And finally, we said that depending, we might expect actually that the DMRT1, that actually a single copy of it would lead to female development, even if you had one wild type copy. So if we think about it, there's this similarity between the alleles that we're talking about, these mutant alleles, and sex chromosomes. And in fact, this is thought to be how new sex chromosomes arise, that the first step in the origins of a sex chromosome is a mutant like this that causes organisms to develop as either males or as females, but without respect to the environment. With respect to the genes, not with respect to the environment. And so we can sort of trace this through a few steps. So let's say that this is the locus of DMRT1 in the genome. We have our 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 pairs of chromosomes. And so what we've said basically is that at this single locus, if DMRT1 is mutant in both copies, so that it's not functional, we expect even across all these chromosomes, this one little site, we expect to see female development. And then we said that even if one of your alleles is a super stimulator, that regardless of your genotype and all these other chromosomes, we might expect male development. Wait, so hold on a second here. Okay, so this is still one site, right? For instance, we would not, if we looked at the karyotype, we would not be able to see that, right? With the karyotype where we see these vastly different X and Y chromosomes or Z and W chromosomes, here we could be talking about a single nucleotide on a 100 million nucleotide chromosome. So we don't expect to have this clear differentiation between X and Y chromosomes, or in fact, there is no differentiation at this beginning stage. But what a lot of really cool genomic work has worked out is that once this occurs, once we have a sex-determining mutation, we tend to see rapid evolution of differences among the chromosomes, such that over fairly short evolutionary times, we start to have visibly different chromosomes, and also chromosomes that are quite different in sequence. And so that this single mutation seems to be the origin, the beginnings of this striking differentiation between X and Y chromosomes, which is to say that this chromosome with the SS we expect to go on to be like a Y chromosome, and this chromosome with the wild type allele, in this case, we expect to go on to be an X chromosome. So we have really witnessed the birth of a new pair of X and Y chromosomes. Okay, so that's what I wanted to say in this video. In the next video, we'll work through particular ways that the observed genotypes in XY and ZW systems might have arisen. But here, let's just summarize and say new sex-determining alleles can rise through mutations in the core sex-determining switch genes, and that new sex-determining alleles on autosomes can lead to the evolution of differentiated XY or ZW chromosome pairs. Thanks very much.